She's going to be presenting on AMR surveillance and monitoring, new technology and changing landscape. She's working with she's working with the salmonella analysis, and she's using pangenomic data, doing a co-research on that, and she has also published a lot of papers regarding these issues. Today's presentation will be regarding AMR surveillance and monitoring, new technology and changing landscapes. Please welcome her with a big round of applause.
Thank you so much for the invitation to be here. It's a real thrill to be able to share some of the work that we've been doing on kind of developing our AMR surveillance. I'm going to talk about the work of a lot of people and a lot of different groups. I may not have answers to your questions, but I'll certainly put you in contact with the right people. So like was mentioned, I'm also from the NARMS system, but I'm based at CDC, so focused on human health. And I wanted to start by giving an overview of the Centers for Disease Control because it is a very confusing place, even when you work there. So this is just as much like a refresher for me as it is an overview for everybody. So we have 10 centers within the CDC. I'm positioned within the National Center for Emerging Zoonotic and Infectious Diseases. And within that center, we have 8 divisions. I'm over there in the Division for Foodborne, Waterborne and Environmental Diseases. And the NARMS surveillance system sits within this division, and it spans 2 different branches, an epidemiology and a laboratory branch. And we do collaborate across those 2 branches. So all the work I'm going to talk about today is based in this division because we're a One Health foodborne surveillance focused division. But I did want to mention we do have a counterpart in the Division for Healthcare Quality and Promotion. I think that's the acronym. This is an antibiotic resistance coordination strategy unit. They are focused much more on the human health, hospital-acquired infection type work. So we do collaborate, but our surveillance systems are very, very separate and actually quite siloed. So Heather did a wonderful job of introducing the NARMS system. It's a tri-agency agreement between FDA, USDA, and CDC. I'm going to elaborate a little bit more on the CDC component and just describe how it really is a very intricate, interactive system that works with a lot of other groups. So we are very connected to PulseNet, our national enteric surveillance system. And PulseNet is tasked with identifying outbreaks and clusters of foodborne illness across the country. They do this by coordinating over 80 different labs across the entire country, working with the state partners to centralize all of the data that comes in. That's the laboratory detection component. We also work with other surveillance systems like FoodNet, which is more on the epidemiology side. So they collect a lot more detailed epi information, like hospitalization or treatments or travel history from the patients that are being infected with these foodborne illnesses. Okay, just a bit of history. So even though they were both born at the same time, NARMS and PulseNet actually functioned quite separately when they started in 1996. PulseNet was focused on DNA fingerprinting using PFGE, and that was done at the state health departments. Whereas NARMS focused entirely on AST, phenotypic testing, that relied on isolates being shipped to CDC to perform that testing. Fast forward 20 years, and we have the introduction of the National Action Plan on Combating Antibiotic-Resistant Bacteria. This was a congressional action plan that came with a budget line that allowed the integration of NARMS and PulseNet systems. What it enabled PulseNet to do was put sequencers in every single state health department and train their staff on how to perform whole genome sequencing. This was a great opportunity for NARMS as well because we were then able to use genotypic resistance to complement our phenotypic data. And now we're in 2024. PulseNet has transitioned completely away from PFGE, is now entirely whole genome sequencing based. NARMS is completely integrated into the PulseNet system. And this has enabled us to expand our collaborations and work a lot more with academic and laboratory partners because we have access to all of this great data. And I tell you what, we do not have the time to analyze it all. So if you're interested in working with this data, please let me know. Oh, an animation I did not realize was there. So how this system works and the integration, all of the foodborne illness pathogens that are reported to PulseNet are nationally notifiable. So this is Listeria, Salmonella, Shigella, sugar toxin producing E. coli, and Vibrio. All of those culture confirmed cases get sequenced at the state, they get genotyped, and they have their AMR determinants predicted as well, and that all gets uploaded to the PulseNet national database as well as NCBI. And that's all centralized. This is like upwards of 80,000 isolates every year that's coming through this system. To complement that, we still have our NARMS phenotypic testing. So 1 in 20 of those confirmed laboratory cases gets physically shipped to CDC for phenotypic testing. So 5% of all of the cases of foodborne illness from these organisms across the country. The one caveat to the 5% is that our typhoidal Salmonella serotypes, so typhi, paratyphi A, and paratyphi C, 100% of those isolates get shipped to CDC. So we have a much more complete data set of genotype and phenotype information for those organisms. We have like 20,000 something isolates that have both phenotype and genotype. It's a really wonderful data set. What do we do with all of that data? Well, first we make it publicly available. So this is the NARMS Now CDC version, which is a different webpage to what Heather introduced. And this has data, WGS data and AST data that is as up-to-date as it can be. The WGS is much more real-time than the AST data. But you can filter and you can also download all of this data by bug, by drug, and by year. So all of this goes into reports. It's also used to inform CLSI and UCAS breakpoints. It is used to inform national treatment guidelines, but it is not used for clinical care. We are not, the predicted resistance is not supposed to be used for patient care. We also report our resistance data in the outbreak reporting. So this is the BEAM dashboard, which is publicly available. It's fairly new, actually. And what you can do with this website is look at the foodborne outbreaks in the US by organism, by vehicle, by exposure, by year, and we're reporting out those outbreaks that have clinically relevant resistance as well in the tiny little print down the bottom. We also put this data into public notices. So whenever we have an ongoing outbreak or investigation, we want to make sure that the public is aware and the public health professionals are aware of what's happening. So we will release these notices that have information about the exposure of the outbreak and also the rates of resistance within the outbreak, confirmed outbreak isolates. So this is predicted phenotype. It's not AST that we put, and that's partially because we have access to more real-time data with the predicted resistance. And we also use it in our alert system. So we have an emerging antimicrobial resistance alert system, the EAR system. And this is a collaborative working group between our NARMS partners, as well as other groups at USDA and FDA and the Public Health Agency of Canada. And we meet monthly. We talk about the numbers that are coming in for the specific alerts that we have ongoing. We talk about whether any action needs to be taken, because if the numbers are increasing, we might want to initiate conversations with the right people to try and do something about that. And we also talk about any new alerts that we want to be introducing. These are things like, a particular gene, MPHA and Salmonella serotype X. I can't actually remember what some of the alerts are. But I have been tasked not to tell you what we're doing currently, but what we are thinking about doing and the new technologies that are on the horizon. And it won't surprise you for me to say that we are looking at long-read sequencing. We've been investing a lot in the last couple of years. And I will say this is because of AMR. I work in the research space, and we needed long-read sequencing to complete our genomes and to study our plasmids. And so I think that initial interest kickstarted the investment in our division for building up long-read sequencing capacity and expertise. One part of this I wanted to mention was the Oxford Nanopore Technology multi-site validation. So this was a collaboration between staff in our division, as well as the Centre for Pathogenomics, which is based in Melbourne, Australia. And the goal of this validation was to do both wet lab and dry lab assessment. So they looked at extraction method and library prep method, and then they also, for the dry lab side, looked at the base caller accuracy. And the wonderful thing about this was that they actually worked with ONT to feed back that information. So all of the assessment results they were finding, they gave back to ONT so that they can continuously improve their algorithms. We want to make sure that long-read sequencing is going to be as accurate as possible if we're trying to use it for AMR surveillance. And one of the things that came out of this was that AMR determinants can be detected at a lower coverage than I think was initially expected. So this is good news if we're trying to develop standardized high throughput protocols. One of the questions of this validation was also the feasibility of using long-read sequencing for global AMR surveillance, and I'll touch on that in a couple of slides. So that work was done by people in our lab, in our team. I also wanted to highlight some of the work that we do collaboratively, which means we give our data to somebody and they do the validation for us, which is a wonderful way to participate. The Global Typhoid Genomics Consortium is a group of researchers, academics, laboratory staff that is being coordinated from around the world, and they have really focused on trying to understand the epidemiology of Salmonella typhi. What they're doing right now is trying to validate the use of long-read sequencing for genotyping and AMR detection in Salmonella typhi. And that is actually just about to be submitted for publication, I think, so it will be publicly available shortly. I wanted to highlight this not because Salmonella typhi is necessarily a foodborne concern for many of us, but because I really believe these global assessments with multiple partners around the world of new technologies and new tools using surveillance data from many national surveillance systems is a really good model for us to be using going forward. So that's why I wanted to point this out. And I'll spend just a few minutes talking about some of the work that I've been involved in around microbial ecology and the ecology of AMR. It's not a new technology per se, but I like to think that it is a new way of thinking about AMR and trying to understand how we got to the situation that we're in right now and maybe predict some of the things that we're going to be experiencing going forward. So when we study AMR in the WGS era, it often looks like this. We build a phylogenetic tree based on core genome targets, and then we map around the outside AMR genes of concern or plasmids. And this is really, really useful. We do it a lot. It gives us good information, but it also sometimes sends us conflicting signals. Like this, here you've got all these kind of random colors around the outside of this tree. Doesn't make a lot of sense. We can't make any kind of good conclusions from this figure. So we are investing in a pan-genome approach, which is using not just core genome material, not just plasmid material, everything in between, the entire pan-genome of these organisms. This is called Jacquard Index Network Analysis. It's a KEMA-based method. I won't go into the technical details, but if you want to ask me, please do. I could talk about it all day. I won't though. So this method allows us to capture both vertical and horizontal evolutionary pathways of these bacteria. So that, to me, is the key way to understand AMR and the evolution of AMR. We also we visualize using a network rather than a tree, which we know is actually more reflective of how bacterial populations evolve, right? So an example of that, we started with Salmonella typhi as the pathogen, and what I'm showing here is a network of over 2,000 typhi genomes. So each grey dot is a genome. It's an individual case of typhoid fever from a patient. And the dots that group closer together are more closely related. And we can do a couple of things from these networks. First, we can study AMR directly by literally mapping on the AMR profiles onto these networks. So you can start to see which profiles are more common in which groups, and which groups don't have resistance. And then we can also link that to our epidemiological data and our geographic data to find out which resistances are more common in certain places. this has been kind of, in my mind, a game changer for understanding, for linking the epidemiological and laboratory data together in sort of a complementary way. We can also study, using the networks, we can also study AMR indirectly by mapping on plasmid information, by mapping on mobile genetic element information. And that will help us understand the dynamics of how these elements move around. They are the elements that are carrying the resistance genes that we care so much about. So understanding their dynamics is key to understanding and maybe potentially predicting where AMR is going. I will say, again, typhi, I know it's not necessarily a foodborne pathogen of concern. We have looked at other Salmonella serotypes that are foodborne, Salmonella hedar, Salmonella enteritidis. So we are definitely applying this approach to understand and to maybe link some of the human cases with the food animal or retail meat cases, which sort of speaks a little bit to the question that was asked earlier. I don't have anything to show for that though. Now, all of this work that I've talked about is happening in a not static environment, right? Our landscapes are always changing. One of the things that has been the biggest change for us recently is our shift away from PulseNet's shift away from a proprietary software for their centralized database to an open access cloud-based system. That's literally happening like as we speak. I think they launched it last week on Monday. And so it is, in theory, going to be a very seamless transition, but I don't want to undersell how big of a deal this is. This is the biggest technological transition that PulseNet has had since the shift from PFGE to whole genome sequencing. So it's a lot of work from a lot of people. What that means for us from the AMR surveillance perspective is that we've moved from ResFinder to AMR Finder Plus, which is a much more up-to-date curated database. It's the one that's in the NCBI pathogen detection model. And so we have all confidence in using this system. All of our internal validation has showed that our results are going to be as expected. But of course, we'll continue to monitor going forward. We always want to make sure we're refining our surveillance system. The other thing that is changing in the landscape of sequencing is that post-COVID, a lot of countries now have access to sequencing that they did not before. But this is not short-read sequencing, it's long-read sequencing, right? And so all of the established national surveillance systems that have developed protocols are based on short-read data. And what we need to do is develop protocols for labs for their long-read data. And so PulseNet International has worked a lot in this space. PulseNet International is a collaborative group of over 80 countries based in 8 regions around the world. And they're really tasked with developing or standing up a WGS-based foodborne surveillance illness system. This is going to look different in different places, obviously. You can't kind of apply the same thing in every place. And so they're really working to coordinate. One of the main things is technology transfer, so developing these standardized protocols that can be implemented in labs. And that multi-site validation for long-read sequencing that I mentioned earlier is part of this process. They are currently, PulseNet International has conducted 2 trainings in the last couple of years, one in the PulseNet Africa region and one in the Asia Pacific region as well, to implement these long-read sequencing protocols. So AMR surveillance globally is rapidly expanding, which I think is a really, really exciting space. And it has been brought up already, but all of these methods are culture-based methods. And what is happening in the US is the rise of culture-independent diagnostic testing, which is wonderful for patient care, but it does mean the sure and slow decline of cultured isolates coming into our surveillance system. And so what PulseNet is doing is piloting a highly multiplexed amplicon sequencing method to extract genomic data directly from stool. They're working with 9 states across the US to kind of validate this. It's promising. We can get detection of the pathogen, detection of the genotype. It's affordable, it's scalable, and it's automatable, which is important for state labs. But we've not yet invested in the AMR component of this. The way to do this is to develop thousands of primers to detect all of the resistance genes that are going to be clinically relevant. It's a big task, it takes a lot of resources, and we are not there yet. And I just wanted to end by saying all of these wonderful AMR surveillance systems cost a lot of money, but they also save money. So an economic evaluation was done in 2016 for the PulseNet system. This was based on PFGE technology. So Even though it was PFG, they found that the PulseNet system prevented up to 270,000 foodborne illnesses every year because they were able to detect outbreaks sooner. And this saved over $500 million in associated medical costs and lost productivity. So this is good value for money. Every dollar spent, we get $70 saved for the US economy. But like I said, this is PFG based. So what we are doing right now, I say we, I'm not involved, is a cost-benefit analysis of the WGS-based PulseNet system to make sure that all of this extra effort that we're putting in is actually paying dividends at the end, right? To make sure we're still spending our money wisely. So look out for that cost-benefit analysis coming soon. And I'll close there. Here's my email address. Please do reach out. I'm always happy to discuss any of this and, like I said, put you in contact with the people that can most speak to all of the work that I've just been describing. Thank you.